much. During the coffee break, I was telling my colleague Timani, who spoke on Monday, that I still feel very shaky when I find myself talking at a vaccine conference. I'm a plant scientist by training. I did my PhD in plant pathology. I still have a very active portfolio on diseases of vegetables and horticultural crops. So please excuse me for my omissions and commissions during this talk, and any questions about diseases of avocado or mango at the end would be very useful. So it was about 7 years back that I got enticed into the world of nanoparticles, especially for vaccine delivery and especially targeting veterinary vaccines. If we look at vaccine development, animal versus human, potential returns for animal vaccine producers are much less than those for human vaccines, maybe surely because of the market size. As a consequence, there is lower investment in research and development in the animal vaccine area. And if you look at the two examples here of Gardasil, market size greater than $1 billion US, maybe much more after I made this slide. And if you look at foot and mouth disease in cattle or mycoplasma pneumonia in pigs, the combined market size is only 10 to 20% of Gardasil. But if we look at the issues in terms of delivery of vaccines, I think whether it's human vaccines or veterinary vaccines, they share sort of a common platform of issues. We have a need for protective immunity, minimization of side effects, ease of handling and administration, cost of production, delivery, and specifically for veterinary vaccines, I think other speakers have also mentioned, because of the large size and the need to give multiple doses, just increases the cost, just increases the problems many folds in terms of delivery and outreach of the vaccines. And another critical factor is, of course, the stability of vaccines as well at room temperature. So when we look at nanoparticles as vaccine delivery system, and over yesterday as well, we heard some very interesting talks on myriad of nanoparticles which are now emerging, both as carriers as well as adjuvants. And I think almost all of them look at these potential advantages, that whether we are looking at additional routes of immunization, we are looking at enhanced immunogenicity, improved storability, and specifically low cost of production. So when I started working in this area with the help of experts in the animal health like Tim Marnie and the nano engineers like Professor Max Liu at the University of Queensland, we started looking at silica nanoparticles as our tool. The first baby steps that we took were with a model protein over albumin, because simply it was available, we could buy it, and then we could do some initial trials on the using silica nanoparticles as the delivery system. Initially, we used MCM41 or amino-functionalized silica nanoparticles. These were about 90 nanometer in size, and the protein loading that we could get was only about 70 micrograms per milligram of nanoparticles. However, still it was encouraging for us, and we did go ahead with a small animal model or a mice trial with 3 subcutaneous injections. Do we have a pointer here? Because of the low loading, we could inject, and this was the first time that we were injecting these silica nanoparticles in mice. We just injected with either 2 micrograms of ova or 10 micrograms of ova, and our positive control group was 50 micrograms of ova with traditional adjuvant quilling. The encouraging thing from this trial was that we could still get a detectable antibody response with 10 as well as 2 micrograms of ova. Not only that, we were also able to get a cell-mediated memory response for the interferon gamma response using these AM41 with 10 microgram of ova injected. Now this was encouraging enough for us that yes, we could inject these silica nanoparticles in mice model, and this, yes, we are getting an antibody as well as a cell-mediated response. We also did then experiments on freeze drying of this ova-albumin loaded AM41 particles using suitable excipients, stored it at room temperature for 2 months, and then did the characterization on the freeze-dried nanovaccine. We used this as well then in a small animal model, and here you can see this is the freeze-dried nano vaccine, and whereas this is the wet nano vaccine, and this is when 50 microgram of ova is injected with quille, which clearly indicates that after freeze drying, the activity of the nano vaccine is still maintained, and we are still getting an antibody response. Not only that, we also got an cell-mediated or memory response with the freeze-dried ova formulation. After this, we moved to a real virus system, encouraged by these results, that let's target not just a model protein like ovoalbumin, but let's shift our focus to a disease called bovine viral diarrhea virus, and for which we can easily say 40 years of vaccination, 160 currently licensed vaccines to eradicate this disease, but apparently not much in terms of control. The disease is still thriving. BVDV infects almost every part of the cattle in terms of ulcers or erosions or neurological defects, and it's also a part of the BRD complex, which Tim Mani gave a talk on Monday. Sorry for showing these gory slides before lunch, but yes, it does infect, can cause death, can cause severe lacerations, and the calves that are born are persistently infected with this disease. Another very critical thing about this disease is that any infection of a fetus between 0 and 125 days of gestation can result in the birth of a persistently infected animal. Not only that, PI animals shed and spread the disease through saliva, nasal secretions, feces, and other discharges, and it can take as little as 1 hour of direct contact with a persistently infected animal to transmit BVDV to an uninfected cow. Pestigard is the current registered vaccine for BVDV and needs to be administered 2 doses of this 4 to 6 weeks apart with an annual booster dose thereafter. It is a pestivirus, it is a single-stranded RNA, the genome size is about 12.3 kilobases, and E2 as the structural protein has been previously shown to be a major immunogenic determinant and has been used in various development of vaccine models. So the first task for us was to express this E2 protein. This was done in E. coli. We first we designed with a E2T1, which is a 93 base pair membrane binding domain, lacks the 93 base pair binding domain, and we also did a E. coli codon optimized E2 expression to increase the yield. As you can see here, that with codon optimized E2, we could get sufficient quantities of E. coli expression to use in animal model systems. Now here we shifted a track from the previously synthesized or tested MCM41 particles because we could not get E2 to load on those particles. Therefore, we moved our focus to what we call amino-functionalized hollow mesoporous silica particles. These are hollow, but had an entrance size of about 2 to 2.5 nanometers. The size of these particles was 120 nanometers, and using these particles, we could get an E2 absorption of 80 micrograms per milligrams of particles. Once again, we did some small animal models, and as you can see here, that we could get an antibody response, and we could also get a cell-mediated response using these hollow mesoporous silica spheres. We also did a sheep trial with these hollow mesoporous silica spheres and got similar results. However, so far, this work had pointed to us 2 major limitations. A, we needed to increase our protein loading. We were not getting a direct comparison with quille, and we were not getting a high enough antibody response. Secondly, if we wanted to move towards a single-dose vaccine, we needed to create some sort of a reservoir in these silica nanospheres or some sort of a depot effect so that we can get a controlled or a sustained release of the antigen over time to reach to our target of a single-dose vaccine. This is when Professor Max Liu at the UQ came with the idea of generating what we call the next generation silica vesicles. This is just a picture of golf balls taken from the net, and this is what exactly these silica nanovesicles are. If you can imagine a golf ball with a very thin wall, with a nice large cavity and having holes on its surface. This is what are these silica vesicles. They are about 50 nanometer in size. As I mentioned, they have a large internal cavity for high payload, and the most important thing is we can control the size of these holes. So we can have these holes as small as 2 nanometer, but we can also have these holes as big as 20 nanometers. Now this facilitates the entry of the protein depending on its size inside the silica vesicles. And these hollow silica vesicles, even the synthesis or the composition step is fairly simple. That you have synthesized it at 2 different temperatures with a surfactant. The first step results in the formation of a vesicular structure, and the second step actually controls the entrance size. And as you can see here, if they are synthesized at 50 degrees centigrade, the pore entrance size is very small. This is the internal cavity, and these small holes marks the entrance pores. At 100 degrees centigrade, we get about 5 nanometer size of these entrance pores. At 140 degrees centigrade, we get more than 18 nanometer size of these pores. And the best part is that we can still maintain the vesicular structure. So they still remain uniformly dispersed, we can still control the vesicular structure, and we have a large internal cavity inside. Now when you think of vaccines, it also gave us another advantage, that by using these silica vesicles, we are injecting minimal amount of silica into the animal. A, the wall thickness is only 6 nanometers. B, it is hollow from inside. So from a regulatory perspective as well, it gives us an advantage when we talk to the regulators as far as the amount of silica being injected into the animal. And also, this provides us with a depot effect where the protein can go inside and can have a slow, sustained release. Another factor which is in favor of these silica vesicles is that it gives them a bit of a fragility. Although we are still doing those mechanistic experiments where we need to show the fate of these particles in a model system, or how they are excreted or how they are degraded, but the structure itself provides those advantages. So using these new generation silica vesicles, we found that the protein loading increased about 3 times, from 80 micrograms to roughly about 250 micrograms per milligrams. So yes, we were on target with increased protein loading. And as with everything else, we did another mice model trial with 3 subcutaneous injections, and here for the first time we were directly able to compare. So all these mice got 50 micrograms of E2 injected with either the traditional adjuvant Quille or bound to the silica vesicles, and you can see here we got a higher antibody response as compared to Quille using these new generation silica vesicles. We also had a group, we thought, how about we have the nanoparticles as self-adjuvants, but we also add Quille to the combination. Will we get even further enhancement of the antibody response? However, that was not the case. But silica vesicles themselves proved to be very nice self-adjuvants. And this was really good to see that the cell-mediated response with the silica vesicle group was much higher as compared to the traditional adjuvant Quille. The next task, as I mentioned, the objective was to move towards a single-dose vaccine. So encouraged by these results, we did another animal model trial where we had 8 mice. 4 of them all 8 mice received only 2 subcutaneous injections. 4 of them we sacrificed at a 3-week time to do the cell-mediated response, and 4 of them were left for 6 months to do the ELISA after every month, as well as to do the cell-mediated response at the end of the 6 months period. Once again, here you can see that after 2 injections, the group 1 and 2 are the Quille groups, group 3 and 4 are the nanovesicle groups. There was a mice-to-mice variation when we have realized now that maybe it's, in order to adjust for this variation, we need to have a little bit, rather than 4 mice, maybe have 6 or 8 mice in group. This always becomes a dilemma when you're writing your ethics application and you're thinking, how to minimize the number of mice and still have statistically relevant data. But once again, there was the similar pattern of antibody response, and after 2 injections and after 6 months post-final immunization, as you can see here, all 4 mice injected with the E2 SV140 formulation gave us a very high and very nice cell-mediated response. So this gives us the confidence that yes, we can move towards a single dose, and yes, we can have a long-term immune response using this silica vesicle nano formulation. These are some of the other studies we did. Once again, in terms of immunohistochemistry for IgG analysis, this is after 3 weeks, and this is after 6 months in case of the SV140 formulation, and this is with the traditional adjuvant quillin. We have also done detailed histopathology studies on almost all our organs harvested from these mice after 6 months, liver, heart, kidney, and we have found, and even the site of injection, and there were no changes observed in these histopathology studies. Not only that, we have also done work on another disease called Anaplasma marginale, which is cattle tick fever, which is again a very, very critical disease causing worldwide huge economic loss and as critical for Queensland as well. And currently there are no safe and efficacious vaccines for Anaplasma marginale. I'm touching on this aspect because this is again shown us another potential for these silica nanovesicles. Now here for the Anaplasma marginale, currently a group at Washington State University, Professor Wendy Brown, they use outer surface membrane proteins as the vaccine candidates. Among those outer surface membrane proteins, it has been shown that Verbi 9.1 and Verbi 9.2, which sit here, can give us protective or at least good immunogenic responses if injected together. So that is, both Verbi 9.1 and Verbi 9.2 may be required as epitope recognition. In this case, what we did was that once again we loaded these silica vesicles. We could get very good loading and we could proceed to an animal model. Here what I would like to highlight is that in this case we injected the mice not only with a single antigen, that is Verbi 9.1 loaded on these silica vesicles, we also injected them with a mixed nano formulation. That is, we loaded Verbi 9.1 on the silica vesicles, we loaded Verbi 9.2 separately, mixed them together, made a mixed nano formulation, and injected them in mice. The idea was, can silica vesicles provide us a platform where we can develop multivalent vaccines, where we do not have to load the antigen on the same particle, but we could load it and then mix together and then inject for ease of delivery. And here you can see that we got a nice antibody response, and we also got a very nice antibody response even when we injected the 2 antigens together, that is 9.1 and 9.2, with no cross-specificity detected, and this is once again the mice injected with 9.2 only or with a combination. And this was also reflected in terms of cell-mediated immunity. These are the 2 Quillay groups, and these are the 2 groups that have been injected with either a single antigen or with a mixed nano formulation. So we can see again that in this case it was SV100. 100 indicates the temperature at which they were made, so the pore size was about, entrance size was 5 nanometers. They can induce high level of cell-mediated response, and we also confirmed that they can be used together as mixed nano formulation and induce high cell-mediated response. Therefore, I think we are at a stage we are still waiting to do large animal model trials or cattle trials as proof for protection or challenge experiments. However, the data generated with the small animal model and with different diseases really show us that we do have a platform in our hand in terms of these silica vesicles, which we can control the particle size, we can do the functionalization, we can control the cavity size, we can control the entrance size, and these can act as excellent reservoirs for loading protein antigens. Not only that, we can do a freeze drying, or they improve the shelf stability of the nanoparticle vaccine formulation. The translational potential is there from animal to human health. We have already started investigating at hepatitis B with a commercial partner, looking at silica vesicles as the delivery platform. So thank you very much. This is the team Nano Vaccines as it is moving forward. We have in the phase 2, we had Washington State University as our partner, as well as funding from Zoetis. And hopefully this work will move more forward, and I'll have some better results to show you whenever we meet next time in life. Thank you.
So thank you very much for this interesting new application. How long would you say the shelf life is of this structure?
So far, we have stored for about 1 year, tested with gel analysis and other methods just to see the protein integrity first, and we are doing now animal models to see its functionality as well. So it just becomes more protected.
Okay. Are there any more questions in the back?
Actually, I'm just wondering, how about the biodegradability and clearance and toxicity of silica?
Yeah, so we have done the cytotoxicity assays in cell lines and compared. I haven't shown the data here, so they are very non-toxic, do not have any effect on cell viability. In terms of biodegradability, one of the PhD students did venture into the arduous task of collecting urine and feces from all the mice injected for 1 week and 1 month. However, because we are injecting such low amount of silica, it became a detection issue. We did go for ICP analysis to see, whether we can detect some silica in passing out, which was not possible. So now we are designing some imaging experiment to see what happens, as I said, to the fate of the particles in the system. But otherwise, in all cell line assays, they are not cytotoxic at all.
So can we get clear from the kidney? So it can be clear from the kidney.
That's what I have to still confirm. That's what I'm saying. Those excretion experiments still didn't give us, because of the low detection limits, we couldn't get a data.
So second part of your talk was modifying the silica, and you said that you had a high loading efficiency from 80 to 300, right?
Yeah.
Yeah. So how do you think that the antigens are released from your silica particle? Because it can have some loss effect and can release slowly.
Yeah.
But you said that it can provide a different effect. What do you
No, exactly. So these are some of very good questions. These are some of the answers that we are looking for as well. In terms of immune response, yes, we have seen that the immune response for a 6-month time period. However, we are designing now assays, as I said, using either labeled proteins or other microscopic structures to see how the release actually happens. But we have seen a distinct difference when we were presenting the protein only on the surface of the antigen to the protein in these silica vesicles. So we have done some electron microscopy densitometry studies as well, just to see whether the protein is present inside as well as on the surface of the antigen. And as far as release is concerned, the hypothesis is that endocytosis happens, and then these silica particles either degrade or the protein comes out. Now, these are the 2 key hypotheses which we still need to work on to get more results. Thank you. Yes. Yes.
Yes, go ahead. Have you tried any bacterial antigens? The ones that you have up there, a viral?
A viral and the rickettsial pathogen, that Anaplasma marginale is a rickettsial pathogen. But so far not bacterial pathogen, but we do have in our arsenal that we would be doing. We have also loaded toxoid on these silica vesicles, but very preliminary stages.
Okay, thank you very much again.